My name is Omri Tomer. I'm a PhD student at Professor Yaniv Asaf's lab at Tel Aviv University, and I'm going to talk about different measurements we conducted in our lab of cortical microstructure in patients with Parkinson's disease as well as in healthy populations using advanced MRI techniques. I'll start with some brief relevant background on Parkinson's disease and the motivation behind this study. So Parkinson's is a disorder that is usually associated with aging, as its onset is heavily correlated with age. If you ask a regular person who is not involved in any field related to medicine or neuroscience what Parkinson's disease is, you'll probably get an answer resembling, it's a disease old people have that affects their movements. And indeed, it is most saliently characterized by motor symptoms, such as tremors, slow movement, and muscle rigidity. But it should be noted that these symptoms are not uncommon in healthy elderly people. Just as a personal anecdote, I recall how in the years before my grandfather passed away, he had all of the symptoms I mentioned above. His doctors described them as Parkinsonism, but noted that due to his old age, they are just as likely to have other causes, and he was not diagnosed as having PD. Now, as far as the underlying pathophysiology is concerned, Parkinson's is primarily associated with the loss of dopaminergic cells in the substantia nigra of the brain, which affect basal ganglia circuitry that is heavily involved in motor functions. So this would be the initial explanation for the motor symptoms, but going back to the earlier comparison with healthy aging, we know that the substantia nigra remains intact, as you can see here in the comparison between a healthy midbrain and a Parkinson's patient midbrain. So the question here, is there a resulting change, for example, in cortical microstructure in Parkinson's that is similar to changes that come with old age and so might explain the similarity in symptoms? In order to examine that question in actual Parkinson's patients, we used an analysis method that was recently developed in our lab that attempts to measure lamination patterns in the cortex with MRI. It does so by identifying different classes of gray matter based on their T1. T1, the longitudinal relaxation constant, is affected by cell morphology, and through the variation of T1 in the cortex, we identify different clusters of gray matter, 6 to be precise. We recently published the methodology for those who are interested in reading more about it. But just to give a brief technical explanation of this methodology, we used multiple inversion recovery scans with a wide range of inversion times, which allows us to calculate multiple T1 components comprising each voxel in the brain. We then use a mixture model to identify the T1 clusters across the whole brain, and specifically gray matter clusters. We then generate for each cluster a whole brain probability map. And you can see these probability maps here, with each row representing a different gray matter T1 cluster. This method has some promise and some limitations, which we described in the recent human brain mapping paper we published about it for those interested. So the question we try to answer in this study can be divided into two parts. The first is whether there are any microstructural changes in the cortex following the onset of Parkinson's. And the second is whether these changes are the same as changes that come with old age, or whether a different cortical mechanism is at play when it comes to Parkinsonian symptoms in both situations. Therefore, this study is divided into two parts. The first, a comparison of the T1 laminar composition of the cortex of Parkinson's to control subjects, and then seeing whether affected areas in T1 clusters are the same as those that are correlated with age in a healthy study population. And so, we used the analysis method I just described with two different study cohorts. For the Parkinson's study, we had a group of 14 subjects in the relatively early stages of the disease, and 14 control subjects of similar age who were scanned with the quantitative T1 protocol I introduced earlier, and underwent the analysis I described. It is important to note here that, as is common in Parkinson's, especially in early stages, the subject had a most affected hemisphere, MAH, and as more subjects had a left MAH, we flipped the brains of those with the right MAH on the right-left axis. We then performed parcel-based morphometry on the T1 cluster maps, using one of Schaefer and Yale's 1000 parcels atlas as the basis to reduce the number of multiple comparisons. For the healthy aging aspect of this study, we used a sample of 200 subjects with a wide variability in age, using the same scanning analysis steps as the first study, and performed parcel-based correlation between T1 cluster maps and age. So as you can see here first in the healthy aging study, there's a strong negative correlation in clusters 4 and 5 in the majority of the cortex between thickness and age, suggesting that these clusters decrease in volume with aging. And there are also some slight correlations in other clusters. On the other hand, when comparing Parkinson's patients to healthy subjects of the same age, there are very few significant differences in the cortex between Parkinson's and controls, but noticeably in the entire left motor cortex, as you can see here, corresponding to the most affected hemisphere I discussed earlier, there are significant differences in the thickness of cluster 3, suggesting that this cluster's volume is reduced following the onset of Parkinson's. So the conclusion we come to is that while both Parkinson's disease and healthy aging can come with similar symptoms, they may be defined by degradation or microstructural changes of different types of gray matter in the cortex. This suggests that while the onset of Parkinson's disease is correlated with age, the disease itself is distinct in its cortical correlates. In recent years, there have been various studies which provided evidence for both possibilities, not necessarily with regards to the cortex specifically. Some show a similarity between Parkinson's disease and healthy aging, while others show distinct differences. We believe our study provides some new insight into this matter, and we are currently working on expanding our study cohort and examining relation to actual motor functions data, and hope to bring more evidence on this matter in the near future. I would like to thank my supervisor, Professor Yaniv Asaf, for giving me the opportunity of working on such cutting-edge research, and also my many collaborators on this project from Tel Aviv University, Asuda Medical Center, Sheba Medical Center, and the Hadassah Medical Center for their help in recruiting patients for this study, especially Don Stein, who helped initiate this project. And thank you all very much for your time.